Hi, everyone. Welcome to another episode of the Modern Biopharma Podcast. We're glad to have Ross Young on today from Biosortia. How are you doing today, Ross?
I'm doing great. It's a pleasure to be here.
Well, we're glad to have you on here today. I think this will be a good episode to learn more about a subject that not everybody knows in my world, but we're becoming a little bit more familiar with it every day. And you seem to be on sort of the bleeding edge of this, so we're interested to hear more about what you do. So, just to get us started, could you talk a little bit more about who you are, what you're doing, and your company?
Terrific. Well, I think you did the introduction. I am Ross Youngs. I am a serial entrepreneur, an inventor, and have a background in engineering and science. But I've been involved in the plastics industry, the medical imaging business. I've been involved in the optical and video disc business. Been involved in things like biofuels. Have won a 6.3 million ARPA Advanced Research Project Award, worked with DARPA previously in the U.S. Air Force Research Lab. So there's a real diversity in my background. And that's really who I am. I've had that opportunity to create products, technologies in my history, and I enjoy it.
That's awesome. Not you don't see a lot of that in the pharma space. We see a lot of people that follow the sort of a typical direction where they're in, say, the chemistry or process side, and they just work their way through. So you've bounced around and done a lot of things. How many companies have you started?
It's nine.
Nine. Whoa.
Yeah. So some of them vary just a little bit, but it's interesting because when you talk about the pharma space and you talk about a lot of industries, I had read a Harvard Business Review study looking at specialists and looking at generalists.
Mm-hmm.
And interestingly, 50% of the innovation comes from specialists. 50% of the innovation comes from generalists.
Wow. Yeah, I actually have a book on my nightstand right now, and it's like The Rise of the Generalist or something like that, and it's sitting there ready for me to read, and I haven't done it, but it was, I think, along those lines. And in the entrepreneurial space, which you're obviously in, there's people that have made huge, leaps in technology that had no business doing it. I think probably the most famous person like that right now is Elon Musk, right? I'm sure you're familiar with Elon. Like, he had no business starting a car company, and I think the first 8 people he had never worked on cars before.
Which is the beautiful way to conduct disruption. Clayton Christensen, may he rest in peace, I believe he passed away in January, wrote the book on disruption. And he talks about how the entrenched always miss. And when you look at Elon Musk, he is the perfect example of somebody that wasn't entrenched and thought that he could do something. The reality of it is any engineering in the automobile industry, any executive in the automobile industry, or investors all would have said he was crazy. And they did until they decided he's not crazy anymore. The same thing is true, and this is one of the fun ones, Steve Jobs. Mm-hmm. When he held up his iPhone 11 years ago, everybody that was in the cellular phone business and their investors were laughing. You got a cell phone that doesn't protect the screen, it didn't flip, and no keyboard. Is he crazy? He has no idea what he's doing. Some of those same companies are going out of business today.
Mm. Wow. So in our space, that definitely feels like that's not allowed, right? In the pharma space, you're not allowed to do that stuff. So I'm interested to hear a little bit more about what you're doing. So you currently your company is in the microbiome space. So it's kind of a generic term a lot of people use. I think the average Joe out there, when they hear microbiome, they think yogurt, kefir, little pills you take if you were on antibiotics because you had an ear infection. So tell us a little bit more about what the microbiome is.
Well, the microbiome is related to a human. We talk a lot about the gut microbiome, which is where the microbes live in a human body in a given location. The reality of it is there's microbiomes all over our body, from our skin to our ears to other parts of our bodies. We have hundreds of microbes. Typically in our gut, 600 to 1,000 different species of microbes, but more microbes than we have cells in the body. So we're dwarfed in numerical numbers of different individual cells of microbes. What's also so interesting about that is the genomics of our own microbiomes dwarf human genomics. We hear all the time 23,000 genes is what makes up the human body. The reality of it is there's a minimum, and they're still trying to figure out this number, of 7 million plus genes from our accompanying microbiome. We can't live without it. It does things like give us metabolites to turn things on, off, up or down in our body. So when you have a hunger pain, or you're craving something, or you have a mood issue, or you have an infection, or you have one of hundreds of other types of things that could be going on in a human body, it is likely related to the microbes that live in you. And I think it was, 2,000 years ago that food was looked at as the most important medicine. Today, quite frankly, eating a good, diverse diet so you have a good, diverse microbiome is really critically important to your health. And trying to feed that microbiome the right things is critical.
Yeah, this is fascinating. The idea that there's millions of genes represented in the actual functioning of your body, rather than thousands of genes, and that should say a lot. And really, historically, say in the last 150 years, when it comes to microbiome or bacteria, most of the focus has probably been on killing it, right?
You're exactly right. And the complexity, the understanding of the impact of that microbiome has only recently surfaced. 25 years ago, we all got told when we figure out the human genome, we're going to know everything we need to know to fix everything. Well, that 23,000 genes is as complex as it is. It's now dwarfed by the complexity of the other organisms that really inhabit our body and are part of the human being.
Yeah, well, thankfully, those bacteria are quite resilient, so we haven't destroyed them all yet, and they're still there, and we can't really escape them. So I'm thankful for that, because I think we probably would have killed them a long time ago. So what I want to move to is, let's talk about your company and what you guys are doing in relation to the microbiome, because there's so many different things that you could be doing. You could be manufacturing stuff. You could be just doing research. So tell us about what it is that your company is doing with the microbiome.
No, I appreciate that. The thing that we're doing is, in a way, the types of things people have been doing for over 60 years. So when we go back into the drug discovery history and we look at successes have come a couple different ways. The vast majority of the drugs on the shelf came from these successes. So 2 examples. Plants have chemicals in them, typically known as metabolites, that can be drug-like, and many drugs came from plants. Interestingly, microbes, which all exist in microbiomes. You don't have a microbe existing just in one place, all on its own, without any interdependency on other organisms or other microbes. So a microbiome exists virtually everywhere. We talked about the human body, but if you go outside and you dig in the soil, or you pull up the roots of a plant, or you go to your closest lake and dip into the water, there's microbiomes everywhere. And what we're doing is what has been historically successful, but with a really big twist. So culturing microbes was the way to collect enough microbes to see what kind of chemistry was there. Now, why microbes? Well, microbes have historically proven to provide directly, indirectly, or inspirationally more than 50% of the drugs on the shelf today.
Mm-hmm.
Culturing microbes is not easy. In fact, just because you take an organism and throw it on a petri dish doesn't mean it's going to grow. And that includes microbes, that includes other things. So less than 1% of all the microbes will grow in culture. So take those two facts together I just stated, more than 50% of the drugs on the shelf came from less than 1% of the microbes that exist.
Mm-hmm.
So what could come from the other 99%?
Right.
That is what's interesting. The other thing that's interesting is microbes themselves put in a isolated culture or an azenic culture will grow that microbe, but you may not turn on any metabolic pathways to elicit a metabolite.
Mm-hmm.
So it may sit in culture and have no need to defend itself against a fungus. Therefore, you won't get that chemistry turned on because no fungus is present. So, there's a number of things that microbes do when they interact. That's why the microbiome is interesting. Because when you have this microbiome, and most microbiomes on the planet have between 500 and 1,000 species that are active in a habitat at a given period of time. That sounds incredibly complex, and it is. Mm-hmm. But the reality of it is they're communicating, they're warring, they're cooperating, they're defending, they're repairing. They're all single cells.
Right.
And where did everything on Earth come from to begin with? We all came from single cells. The first microbiome on the planet evolved and created everything that exists today and every microbiome that exists. So what's wild is our gut microbiome overlaps genetically and metabolically with an ocean's microbiome. If you go to an ocean and you pull out a sample to look at that microbiome's genes and metabolic pathways, it's a 73% plus overlap to the human microbiome.
Yeah.
So what we're doing is we're going back to what was the most successful approach of drug discovery. We're collecting organisms at the kind of scale nobody has ever been able to do for a variety of reasons. And then we're going through the technologies that have existed in the past and the advancing technologies that exist today to slice and dice this material to get at those hidden metabolites. That's what we do. Many other people that are focused on the microbiome itself are looking at bugs as a potentially therapeutic alternative or bugs as a probiotic. We're going to the chemistry of microbiomes to pull out the drug-like molecules that already hit targets and have been basically evolutionarily prepared to be drug-like molecules. Mm-hmm. And we do this with a technology that allows us to grab all these microbes rapidly without destroying them or lysing the cells, preserving them so we then can take it into the lab and slice and dice it. And that's really what Biosortia is doing. We are mining, we are prospecting, we have an industrial scale capability to grab living microbiomes to look so deep into the hidden chemistry. And it's that hidden chemistry that matters. The peaks that exist in all chemistry, everyone can see. But what is that chemistry that we don't see?
So how are you able to isolate that? So when this is becoming more clear as we're talking, so this is great. But I can see how this plays out on paper, sort of. But how are you able to isolate, what was it that actually caused that bacteria to do what it just did, if it's not very isolated, right? So if there's this one and then there's 8 of them around, right, and it's responding differently to all of them, how are you able to figure out which it's responding to? Because that's ultimately what you're trying to do, right? You're trying to go, hey, if you put this all by itself, it's going to do something, and we're going to figure out what it did and go, that could be useful. Now you put it in a different environment with all these other ones, it's doing a whole bunch of other stuff that we didn't even understand, have access to. How are you able to figure out what the one was for the, given sort of scenario, and then maybe be able to use that from a replication standpoint?
Yeah, that goes back as in drug discovery, whether you go back even 100 years. If you found an activity in something and you were trying to figure out what would end up being that medicine, let's use penicillin as an example. It was a fungus that was in contact with a bacteria, and the bacteria couldn't grow into the penicillin fungus. So why couldn't it? Well, in studying it, they ultimately got to that chemistry that was causing that bacteria inability to grow. It was an antibiotic. So everything is based on activity. So what we do is we obtain the biomass. And typically, we're not lab scale, we're industrial scale. So someone may say a lab scale is a liter of something, and then they can look in that liter, and because you've got 1 liter of material, something in there in very small quantities you may miss or may not even show up. We typically start with 20 million liters.
That's big.
Which is orders of magnitude way beyond the scale that anyone has ever attempted. And it's not an attempt on our part. We can do this every day we choose to do this.
Okay.
So we start with 20 million liters, obtain a microbiome from the aquatic environment. Why do we love the aquatic environment? Well, it was the origin of all life on this planet. It has an incredible array of diversity and overlap to every other organism and every other habitat. Why is its habitat so rich? Well, when you start to thinking about variables, you've got things like temperature, oxygen content, pH, pressure, sunlight, nutrient level. You can go on and on and on. There is a habitat in the aquatic environment, whether it's salt or freshwater, which is another variable, where microbes live in microbiomes, and they all produce different types of metabolites, and they're all different types of organisms. What we like to point out is nature is really conservative. If it wasn't conservative, we would have an endless supply of chemistry to play with. But we don't. We've had so little chemistry to play with that we've had to go to creating chemistry with chemistry kits and try to play with that chemistry. But give you an example. When we talk about humans, we all have different DNA. We all have different microbiomes. But our chemistry, the vast majority of our chemistry, the kind of molecules that circulate in our body, they may be at different concentrations, but they're pretty much the same. So there is this finite existence that nature has honed in on of bioactive small molecules that are the best drug-like molecules for hitting targets. That's where we focus. And as we get that biomass, we start to slice and dice it through extractions. So we can separate out the proteins from the lipids to obtain the small molecules. And then we take those small molecules in a bunch, and then we run that through fractionation, where we start to slice it even more. when we end up with highly, highly refined fractions, and we may have hundreds of these or thousands of these, we can then take those to assays.
Mm-hmm.
And in the process of hitting an assay, we find the activity of that little slice of chemistry. Then we will take that back in, isolate, purify, and retest. In that process of isolating and purifying, we end up with a couple dozen more isolated compounds. The reality is there's such a wealth of accessible, unknown, drug-like chemistry that exists in microbiomes that has gone totally untapped.
Mm-hmm.
And that, we believe 10 years from now, there will hardly be a life science company that isn't trying to figure out how to prospect microbiomes. And what we'll end up doing in humanity is we will narrow in on that list of actual bioactive small molecules, and it will be a real dramatic improvement in hitting the life science, whether it's therapeutics, whether it's agrochemical, whether it's cosmetics, whether it's longevity, or even sustainability. We will get to the kind of chemistry of life that we've never had access to.
Right. So in my world, where I see a lot of the new companies coming out are in cell and gene therapy is very popular right now. It's kind of the it's not really new. It's been going on for a long time. It's just finally hitting its stride and becoming a little bit more scalable. So that's right now big, and all kinds of companies are starting to do that. This seems like it's, I don't know, it's probably connected in some degree, but really you're working on this whole other genetic space, right, with the microbiome. So this would be another layer that we're going to stack on top.
Well, and I think that if you really tried to draw the chart and envision the future,
mm-hmm.
and I think that that's a tough thing for people to do. They'll center on the topic that they best know about. So if it's gene therapy, they're going to center on gene therapy, and then there will be tools surrounding it.
Right.
I actually believe the core chemistry of life should be the center, and everything else should be a tool to utilize it. Because even if you find the right molecule in nature to hit a target, there's going to be the opportunity to improve it, the opportunity to deliver it, the opportunity to modify it in some way. And all these other technologies are brilliant technologies. They're not useless technologies by any means,
right.
But they're technologies of the infinite. And what do I mean by the infinite? Well, we already talked about every human has a different set of genes and a different
mm-hmm.
set of genes within the microbiome. And if you play with the genes and you play with that to get to something, you've got an infinite number of opportunities. But if you look at the bioactive small molecules, the cell-to-cell signaling molecules, that's actually a finite set. And it's absolutely critically imperative that you understand as much of the variables as possible. And when you're dealing with the infinite, you've got an infinite set of variables. So you've got to first truly understand what's missing. So I'll give you an example. In the body, there's something going on that turns on Alzheimer's.
Yep.
Billions and billions of dollars for decades have been spent trying to figure that out. There's probably molecules doing something inside the human body that turn that up. Why can't we see them? Mm-hmm. Well, we don't have the capability, the technology to see all the molecules that exist in human, because even if you took 1,000 humans, like I do biomass, sounds really awful, deconvoluted them and looked at all the chemistry, you still may not find molecules that reside in the human body at picomolar levels. So you need to have a source of this chemistry so you can find this molecule and then identify the molecule in a patient that has that activity. So whether you call it a chem marker or a biomarker, the reality of it is the vast majority of the understanding we have in the chemistry in the human didn't occur from slicing and dicing a human. It came from outside the human, from other understandings. And that's how we'll progress in filling in the missing gaps of molecules in a human. And there are tens of thousands of those molecules we can't identify because they're just blips in a mass spec. It's just, you can't even see what it is. Well, if you had more of it, you could see it.
Okay.
And identify it. Identifying it's the most important thing.
Right. So as a company, how much of that is what you are doing versus, I mean, are you turning this over to other life sciences company? Are you doing a lot of this identification on the front end, or what's, I guess, where does your company come into play aside from, harvesting and mining? Are you guys doing the mining to understand, or are you guys doing some mining and then letting everybody else do that?
So where we had to get, we started out and talk about in the entrepreneurial world, pivots are not uncommon. We started out with a need, and the need was for a sustainable source of bioplastics. Okay. How'd that get to therapeutics?
Yeah.
It got to therapeutics because one of the sustainable sources of bioplastics, because you could take all the crop waste in the world and that price will go nothing but up as you move it from place to place. So even though bioplastics are being made and they're possible, the raw materials for the bioplastics are increasing in cost. So we looked at eutrophic waters or polluted waters where there are microbiomes, where there are organisms growing rapidly because of all the conditions to cause that growth. And we started to say, what if we use that for bioplastics? Well, the first problem we had to solve was how do you get the biomass out of the water at an economically effective cost? Because you can't centrifuge a whole lake. It would cost way too much. And you couldn't use chemistry for it. So how do you get it out effectively? Especially when you have maybe 0.05 grams per liter in a very green lake. It's costly. You got to handle a lot of water to get a very little bit of biomass. And that was the Department of Energy's biggest issue. And we started to play with concepts around that, and it came up with a technology that did it at very low energy. What we learned about doing it at low energy, we were also preserving the cells. So as we worked through that program, we ended up pivoting toward drug discovery because so many drugs came from culturable microbes, and there was this vast resource now of unculturable microbes we could tap and explore. So we advanced the technology, proved the technology, we're completely at scale, and now we're sitting paused. Why in the world would we be paused? We're paused for the reality of how it is hard to do disruption in an entrenched industry. Think about what they're teaching at Harvard, what they're teaching at every other university when it comes to drug discovery. How many are teaching deep accumulation of microbiomes and then deconvolution? Anywhere in the world, tell me somebody that's teaching or working on that. Is the NIH working on it? Is there one pharma company in the world working on it? And the answer is nobody.
Yeah.
So just like you mentioned Elon Musk at the start of this, I'm going to guess you didn't know this. Do you know when the first electric car was proven?
The first of his or period?
Period.
Oh, I don't know. Probably 80 years ago or something. I have no idea.
Get ready to fall off your seat. It was 1835.
Holy moly.
Now imagine if in 1835 they continued to work on and perfect the electric car.
Right.
Where would we have been today?
Right.
But that's the challenge with disruption. Everyone has been trained to do everything else. Billions of dollars are going into these infinite approaches to the finite. And we are saying, at an industrial scale, which has never been done, we can go get that chemistry. And we're at day one. Imagine if we pursued this for 5 years, how great this technology could advance.
Mm-hmm.
That's the reality about playing with something. But if you're not doing what's in vogue, you're out of the loop. And when you have so many people trained to do this, that's where the focus is. And that's the biggest issue for what we're doing, is I hope we don't become the electric car of the past and die.
Yeah.
If we die, will it take 100 years to figure out that this chemistry is there for the taking?
Yeah. So I'm just something's popping right now in my head, but I mean, is the availability of, say, computers and how much computers have advanced in, say, the last 20 years, all this, the way we approach things for the most part now is based on sort of our ability to even look into things, right? So if your microscope only goes to 1 micron, you're not going to go down below that in search of stuff because your microscope doesn't work that way. If you have this much computing power and you need this much to do something, you're kind of stuck, right? So I'm just wondering if maybe this seems like it would involve a lot of variables, right? As we're talking about the infinite. So maybe is that helpful in this regard from a mining what's possible in this world? Just the availability of computing power now that we never had in the past?
Yeah, I think there's a big fallacy going on in the computing world as a solution.
Okay.
And I think it's a shame. But when you look at the wealth created over the last 30 years and you ask Bill Gates, how do you solve a problem? Computing power.
Right.
Okay. So what will his focus be?
Yeah.
And now you go look at the vast array of tech entrepreneurs that have made billions in that world. We have put an overemphasis on computing power as the end all and solve all. When it's an infinite problem, you cannot calculate your way out of it. And it is ignorant to think you can.
Okay.
So I'll give you one example. Back in the early 60s, we pursued, as an industry, combinational chemistry. Small molecules was the basic target of CombiChem. But depending on who you talk to, because of spin and orientation of these small molecules, but you take the elements and you put them in combinations, and even if you tried to do that, the numbers of small molecules under 500 molecular weight is so extensive, it's way more than the atoms in the entire known universe. And this is not an infinite problem. This is a finite problem, meaning that if you did a billion tests per second for the next 8 billion years, you would still be at 0.0, followed by more than 100 zeros, 1%. Now, if you take the complexity of chemistry, you need to have more knowns. You need to understand what molecules are made. if you're doing this in the blind, you're doing it in the blind, and you can calculate this till the end of history.
Yeah.
And that's one of the problems we have with computing power is people that are focused here have their solution here. And I hear this. I had a conversation with a leader of a research portion of a major pharmaceutical company. And I asked him specifically the question about generalist versus specialist. And he considered himself a generalist.
Mm-hmm.
He's a manager. He's a professional manager. A professional manager that understands management does not make a generalist.
Mm-hmm.
And I think that that's one of the distinctions that people fail to understand is that you can have all the professional management in the world. That does not make them a generalist. Yeah. It takes the ability to understand a wide variety of technical, scientific understandings and put it in a context that's relatable. Mm-hmm. And I think that if you're focused on computers, that will be where your focus remains. Right. And I look today at computational biology as being the next shiny object that's going to solve all the problems. But that comes on the heels of CombiChem, that comes on the heels of synthetic genomics, that comes on the heels of every other technology that wasn't looking at the available information. And it's that available information that's key. You look at the human metabolome database, and there's less than 50,000 molecules in the human metabolome database. It's representing just the smallest of what's really there. Why not double that number? Mm-hmm. Why not quadruple that number and then plug that into the computers? Well, the problem is now you have more variables for the computers. Mm-hmm. Right. So you need even more calculating power.
Right.
So their tools, they're not going to be the solution.
Right. That's really interesting. Yeah. And the next tool is just another step on the same ladder. And I guess you're kind of saying there's another ladder over here we could try.
Well, and the thing is, if you have a problem, you want to know the variables.
Yeah.
All right, and that's in every field. If you're going to build a building, you need to have the variables. If you're going to try to build the building and not have any variables to plug even into the computer software you need, you're going to have a problem. And I think the known world is accessible in a way we thought it wasn't. And we started to train people 40 and 50 years ago. And there was this shift away from what was working. Mm-hmm. Now think about that. Why did it shift from what was working? We're an impatient society and we're impatient investors. So you see a shiny new object, you throw money at it. And then when that doesn't work, you look for another new shiny object. And the reality of it is what was working, if we had even dedicated a fraction of what has been spent on what was working over the last 50 years, we probably would be in much better shape. Mm-hmm. But we like the shiny objects.
Yeah. Well, sometimes that immediate success is that's what we want, right? And that immediate success keeps us from other successes that might have been better.
Well, and the immediate success for many is pretty powerful because if you come up with that shiny object and you can sell that shiny object, you now have made your ROI, right? And you get to go, great, now I'll go do something else. Right. And I think that that's one of the challenges with the technologies that have come on and the billions and billions that have been invested and the billions that have been used to acquire. They still need to measure success. And they got to be measuring success by the right set of measurements. And I think that that's one of the challenges is if you look at the 50,000 molecules, and quite frankly, many of those have not been identified in a human, they're believed to be in a human. Mm. Okay? So you look at those 50,000 molecules, and then you take all the dollars that have been spent in the last 60 years on drug discovery. finding one molecule to add to the human metabolome database should be worth something.
Mm-hmm.
Now, we have created a lot of molecules that are unnatural in the search and the pursuit of drug-like activities.
Yeah.
Nature's made these things. They've got 4.2 billion years of biology making these molecules, and they are there for the picking. It was believed to be impossible. It was believed to be too hard to go get them. It's not. They're there.
Yeah. Yeah. Wow. Okay. So, man, this is fun. I could actually keep talking to you about this because it's you have a lot of good energy about this whole thing. But we can't go for forever, right? We talked about this at the beginning. One, I guess, just to sort of zero back in on maybe some potentials that you're excited about. So, what are some of the things, maybe specifically, that you see as high potential, good potential in the research, using this as sort of the backbone, I guess, of it?
Yeah.
In drug discovery.
I think if we were adequately funded, we would really look at the immune system. We would look at how to modulate the cytokines that we do know are there, whether it's something that would activate or turn them off or modulate them somehow. So that would be one of our first areas to go do this research. The problem is our company spent so much money in building and getting and proving the technology.
Mm-hmm.
And we're so new that it's really hard for dollars to come our way. Very, very hard for dollars to come our way. Just the fact we want to do it doesn't mean we can do it. And that's the challenge. When we look at next targets, we think neuro-CNS are brilliant targets for what comes from microbiomes. So when you start to look at all the diseases that relate to neuro-CNS, we think that the kind of materials that come out of our source would be ideal. Another one, and we wrote a little paper up on this, I think in March of this year, antivirals. The reality of it is a number of antivirals came from these sources. Well, if you have the capability to prospect these sources at a level never believed possible before, you should be able to find new antivirals. And the same thing with the anti-infective spaces. We hear a lot about drug-resistant microbes. Well, quite frankly, nature has had to deal with that for 4.2 billion years and has that chemistry in place. So going to that chemistry and finding new anti-infectives is highly probable as well. So we think there is a wealth of opportunities just in the therapeutic space. In the agrochemical space, there is a wealth of opportunity as well.
Right. Yeah. Obviously, that this has potential in almost every direction, I would assume. So,
and our major goal, when you said, what do we want to do? We want to take the technology we already have in place and execute. And right now, what we need to have is we need to have a scale to execute that people don't have. Again, when you think about what people are doing when they're culturing microbes, they're dealing with 100 to 200 grams of material. If it's a very large culture and they have isolated and purified the biomass to get it out. And we're dealing with what is 1 million grams of biomass from one single harvest.
Right.
So everything we need is off the shelf. It exists. It just needs to be put into what we're describing as the industrial deconvolution process so that we can slice and dice efficiently. With that, we will build a massive library of unknown metabolites from microbes that we'll be able to share with researchers, we'll be able to share directly with our sponsors or go after targets for our sponsors. But that will be our major goal, is to take those molecules. Now, the interesting thing that occurs by us executing, we also obtain known but rare molecules. So what does that mean? Well, even in the human metabolome database, there's molecules that people know they're there, but they have no idea what they do.
Mm-hmm.
They don't have enough of it to actually even test and screening. Well, in the process of what we do, we obtain massive quantities of peak metabolites that other people have never had access to because it's so prohibitively expensive.
Mm-hmm.
So by building and executing, we end up with a library of molecules available to do the work we're trying to do.
Wow. Well, it seems like the potential for this is pretty amazing. So, who are the kind of people that you're wanting to connect with, I guess, in the world, or who would you want to reach out to you, if this what goes out to the pharma world or the research world? Yeah, who do you want to be calling you back after this?
Well, I would love to have people with dollars.
Yeah.
People with vision. So what we started to do is we started to go to the VCs, we started to go to the pharma companies. Well, what model do they use? They have had such limited success in their own research and their own acquiring of technologies or platforms that it's cost them ridiculous dollars. So they have taken the model of bring me an advanced molecule that is really ready to go into the clinic. Then we'll talk and we'll buy that molecule. Mm-hmm. So we have got a ready set group of customers if we get to execute, but they don't want to be the customer of the platform. Mm-hmm. They don't want to invest in the platform. So what we've been trying to do is we've been trying to go after very high net worth individuals, thought leaders, technology leaders, because we think we can explain them to this and they can take a flyer on it. Mm-hmm. We don't think it's a flyer. We think it is absolutely the way to go. And in time, everyone will follow this path. Now, will they make it their lead program? Probably not. But everyone will have a component of mining microbiomes or prospecting microbiomes someday in the future. And the one thing I've got to say is we know that prospecting natural habitats is somewhat of a dirty word in this industry because it sounds destructive.
Yeah.
Ours is non-destructive and non-impactful to any habitat or environment. It is so benign. We've been approved, and actually done the harvest in a marine sanctuary, getting state, local, federal approval in California. Mm-hmm. What we do
in California.
What we do is benign. Yeah. The other advantage of doing this in the US is we're not parties, whether fortunately or unfortunately, we're not parties to the biodiversity treaties that exist. So when we agree with a source site, we end up owning 100% of what we find, which is an advantage in the US. So I just wanted to make sure that your listeners understood that this is the most environmental friendly prospecting on the planet. And when we go to a body of water, 20 million liters sounds like a lot. But if you looked out at a 10-acre lake, which is a pretty small body of water, it's just a fraction of that water.
Right. Yeah, no. When you were describing what it was, again, it sounds big in our world, but in reality,
at a lab bench scale, it's massive.
Right. Yeah, but I mean, if somebody's mining any natural resource, they're doing far greater, damage or disruption to that space than you are. So. Makes a ton of sense. Well, hey, Russ, this was fascinating. I thoroughly enjoyed this. I learned a lot today. This was not a typical episode, so I'm really glad I got to have you on today. I'm going to put in the show notes things about your company, about you. So if somebody wants to reach out to you or look you up, they'll be able to find you very easily. And, people will be able to see this on YouTube, on LinkedIn, and on any other podcast platform. But I just want to say thank you for being on today. This was great. I learned a ton. I hope and wish for you massive success in this, because I think this seems like a really neat technology that our space could really use. So thank you.
Thank you.
All right.